It's just think it's possible. So I'm a geologist who dabbles in microbiology. So come with what geomicrobiology. A lot of the stuff I work on is evolution of early life and how we can figure things out from the rock record. So is anybody here got any geology experience at all? Okay, so at least five of you will know what I'm talking about, and you'll do all right on the exam. Um, But no, I'm just going to talk about it from looking at rocks, trying to infer something from microbiology. I'm not a microbiologist. I'm not a molecular microbiologist in particular, even though a lot of my colleagues are. So when we think about evidence of life in deep time, we usually look for physical records, right? Things such as rocks, or we use the molecular record, what's in modern biology. So like I say, I'm going to focus on the rock record. Now, I'm guessing this is a figure many you've never seen before, right? Okay. So what it essentially shows is the evolution of oxygen in Earth's atmosphere through time. So remind me, who produces oxygen today? Well, cyanobacteria, right? Out of curiosity, when do plants evolve? How about plants, right? 400 million years ago, something like that. Okay. So animals around 700 million years ago. So the world was dominated by microbes. Okay. And cyanobacteria are more evolved than a lot of other ones. So they're not even the first ones, but essentially we owe our existence to them. Yeah. Because the O2 they produce oxygenates the earth, which oxygenates the atmosphere, which means then that eukaryotes can evolve. What this essentially shows is the evolution of O2, not cyanobacteria. Those are two different things. The cyanobacteria produce the O2. What I'm showing you is the physical record of O2. So what this is showing, so these terms may mean nothing to you, but I will just out of habit talk about the Archean and Proterozoic. These are two eons in geological time. This is basically from 4 billion to 2.5, and this is 2.5 to 500. The Phanerozoic's what we're in now. This is the age of animals and plants. This from here all the way this way is microbial. Just out of curiosity, when did microbes evolve? Any idea? Like how long has Earth had life? Someone just yell out a number. 4 billion. Yep. 4 billion is a good guess. We don't know for sure, but certainly by 3.5, we have evidence for it, and we just based on what we think we have evidence for, we just kind of push it back. We don't really know. And the reason we don't really know is we don't have sedimentary rocks until about 3.8. Sedimentary rocks basically are rocks that are formed from rivers washing things like sand and silt and mud into the oceans, right? So sedimentary rocks are important because if you find an ancient sedimentary rock, you know you had ancient water. You need water for life. So at least at 3.8, we already had stuff. So we just, four is just a guess. So going back in time to four, basically we have very little oxygen in the atmosphere. No biotic oxygen at all. This is completely unknown. There is no way of knowing what the number is other than it's super, super low. Then around 3 billion years ago, we think cyanobacteria evolved, and I'll get into that in a bit later. And then you see the oxygen is up to this level right here. This level, if you track it all the way over to the y-axis here, is percent atmospheric levels, right? So that's 100%, 10%, 1%. This is all the way down to 10 to negative 6 pounds, so a really low level. That level, if you're wondering how do we know what that is, it comes from this thing right over here, and this is a sulfur isotope record. I'm sure you're all familiar with stable isotopes, stable carbon, sulfur, nitrogen, stuff like that, right? So in terms of sulfur, you're probably familiar with 32, 34, but there's also one called 33. Now, what is really unique about this, and this will probably be testable. If I were to have to ask, multiple choice final, it'll be like, what is the evidence for O2? You might say the sulfur myth record. Okay, here's what's important. To get this signature 33, you basically need to have no oxygen in the atmosphere. Because what happens is a volcano puts sulfur dioxide into the atmosphere. It gets zapped by basically ultraviolet radiation, UVC, around 200 nanometers. That converts the SO2 into elemental sulfur and sulfate, and it gives it this 33 signature. So when you look at the rock records and you see 0.5, this is different. All the red represents the sulfur that is to be positive or negative. This tells you, this is to be the smoking gun for O2 in the atmosphere, is there's no oxygen. Because if there's oxygen, you get ozone. If you get ozone, you can't have a photochemical reaction. So this sulfur MIF to me is the best record for O2. Now that translates into this number of 10 to the negative 6% atmospheric levels, because that is the concentration of oxygen at which you start to break down that sulfur signal. If you have O2 above this, there is no sulfur signal. So the fact that we have this signal tells us we had O2 less than that amount right here. Now, the area and time that I work on the most is this thing called the great oxidation event. This is at 2.5. This is the point in time when oxygen in the atmosphere accumulates to the point that there's a significant amount there. Another testable question, when is the great oxidation event? This is, to me, probably one of the most important time frames in Earth's history, because it's when we suddenly have oxygen in measurable amounts. So oxygen then goes up during this thing called the great oxidation event, and then it can do one of either two things, and this is all dependent on how we interpret the rock record. Many suggest it just stays at this level of 1% or 0.1% O2 for a very, very long time. We call this the boring billion, until we get into the Neoproterozoic. Has everyone heard of the snowball Earth? When the planet was completely glaciated between 700 and 600 million years? Okay, that is this period in time. After the snow melts, we suddenly get the evolution of animals. And eukaryotes evolved beforehand, but they really, basically, the green algae, the red algae go crazy after this point. Big question about why deglaciation would lead to animal evolution, but that's the case. And then we get all the way up to here, we get the next jump in O2, and this is plants. Okay? So the very first one's a great oxidation event. This is your Neoproterozoic one, and this is plant plants. I'm just going to focus our talk on here, okay? So that's interpretation one. Interpretation two is that O2 can jump actually at one point in time, almost to modern levels, and then it drops. And I'll explain that as well. But of all the things we know very little going back older than 2.5. Now, here's the fundamental problem that we have. We're ever trying to interpret the evolution of life or what metabolism came first. We don't have enough rocks to look at. Here's the thing. Over time, rocks form. Think of the Rockies, right? They're rising, they're getting bigger. Then they get weathered, right? So they break down. So at some point in time, there's going to be no rocky mountains. It's just going to be flat. Throughout history, mountains form, mountains erode. Okay? Then they get washed into the ocean, some of the sediments, and eventually they get subducted and put back into the deep mantle. In other words, there's very little preservation of rocks older than about 200 million years. Of course, there is, but most rocks have been destroyed. I can tell you there's only 3 rocks that we know of, sedimentary rocks, that are 3.8 billion years old. They're in Greenland and 2 are in Canada, and these are sedimentary rocks. Okay, this is not stuff I'm going to test, and this is just stuff to know, right? So here's the problem. If I want to look at what was life like at 3.8 billion years ago, I got 3 places on Earth I can go to take a look at a rock. Now, those rocks have also been buried kilometers. They've been heated up to temperatures like 800 degrees. So if you were a cell of bacteria, at 800 degrees, you're gone. You're no longer there. So your physical traces are pretty limited, right? So this is kind of the problem we have as geologists going back in time, because there's very few things we can actually look at to tell us anything, okay? The molecular rock record is even more problematic, because although you can take a look at extant cells and you can say, okay, well, this one came from that and this is the deepest branching, put a time on it, you have to use some kind of a geological marker to say that belongs to this time, that belongs to that time, right? Or else you can't track your mutations. So the little data as geologists that we have, we share with you, and then you come up with molecular clocks, and this is how it goes. So this is why there's so much uncertainty. But what I can say that we do know about the great oxidation event is there actually is evidence in rocks that's real. This is, like I said, the smoking gun. But some other pieces of evidence. This is a rock called a conglomerate. It's basically just big pebbles with sand, and so something carried by a river, dumped right basically at the river mouth. In it, you often find rounded grains. Go to the North Saskatchewan, you find these big rounded grains too, right? Tells you it's been bounced around for a long period of time. Now, here's the point. These rounded grains are made up of things like pyrite. Now, pyrite's fooled gold, which is a reduced form of iron. In other words, it's ferrous iron and sulfide. versus ferric iron, which is rust. Okay, pyrite oxidizes to rust. So here's the thing. These rocks at 2.5 and older, these conglomerates have rounded grains of pyrite, but we know in the presence of oxygen, pyrite's unstable. So how do you have a grain that's been rounded, which tells me it's been transported for a long time from the source all the way to the mouth of the river, yet it still exists? Well, the only way to argue that is that there was no oxygen to oxidize it. So this is one of the pieces of evidence that before 2.5, there's no oxygen because these things are preserved. After about 2.5, we start getting these really rusty colored sandstones and rocks. These are called red beds and paleosols. Now, a paleosol is just basically an ancient soil. Now, imagine there's no oxygen and you've got a rock forming into a soil. Okay, let's take Hawaii as a great example. So I'm thinking about the mafic lava, the dark lava. That's kind of what early Earth looked like. A lot of like Hawaii's. In the absence of oxygen, that rock dissolves because of water falling on it, right? The iron in it dissolves out and gets carried away. The second you get oxygen around, that iron gets oxidized, turns to rust on the actual particles. So basically, the rusting of soils is evidence of O2. We don't get that until about 2.4. And although there's lots of pieces of evidence, another one are these manganese oxide ores that form around 2.4. You can't form a manganese oxide, MnO2, without the presence of O2 to oxidize manganese 2. In other words, I would wager that this is true because there's lots of evidence in the rock record that say there was no oxygen, now suddenly there is oxygen. Okay? So this part's true. This part, though, is all kind of guesswork. Now, this is more for the community I was talking to, but this is just a couple other points I want to make. That sulfur MIF record, remember what I was showing you right here, where this is the original work was by a guy called James Farquhar, who was actually a U of A grad, who came up with this paper. He had it stop at 2.5. So we thought the great oxidation event begins at 2.5, and there's like no oxygen, suddenly there's oxygen. All right. Recent work has been looking at rocks. This is the sulfur MIF, just different colors. This is old work, this is new work, showing all the way to about 2.2, we get this. The take-home message is this. So don't worry about the details. It used to be no oxygen, suddenly there's oxygen, and then we stay oxygen. Now what we think happens is oxygen gets in the atmosphere, but it goes up and down. It oscillates up and down until eventually at some point it stabilizes. Okay, so the very first time oxygen went in there, it might have been oxidizing, and it drops out, it goes in and drops out. In other words, it was a non-steady state for a long period of time. And you might be wondering, why would that be the case? These blue lines represent glaciation. So I talked about the snowball Earth event that most people know about. This is the stuff that happens around 700. What a lot of people don't know is there were also global glaciations around 2.4 billion years ago. So imagine you're a cyanobacteria, you're growing on a beach. You're super happy, right? It's nice and tropical. Now you're in the Arctic. Not so happy. So you can imagine why things fluctuate up and down, right? Because as soon as life gets established, a glaciation comes along and wipes out a lot of them. Some survive. It takes a while for them to get back, put more O2 in. Next glaciation comes. So there's these glaciations that are coming in, basically throttling the communities that exist at that time. And it's only after this glaciation that we don't see another glaciation until 700 million years ago. So that all makes sense. That's why the O2 goes up and down. But to reiterate, in terms of testable things, the great oxidation event, the evidence for it, when it was, are the things I kind of want you to get out of this. Okay. And the last thing I want to talk about on this slide is this thing called the Lomagundi event. It's 160 million years when you have this really weird carbon isotopic excursion. Is everyone familiar with the carbon isotope system, like photosynthesis takes in carbon-12, leaves behind carbon-13? Okay, so imagine this. If you have a lot of primary productivity, so a lot of plankton, they're sucking up the carbon-12 into their biomass. They die, they take the carbon-12 with them, right? That means the fluid left behind is carbon-13 rich. At that point in time, any limestone, any carbonate minerals that form are going to be characterized by the positive. What the argument is that for 160 million years after the great oxidation event, there's so much high primary productivity that so much organic carbon is being buried that's why you get this excursion. And it's an excursion because an excursion means it's unique, After 2.6, it stays flatlined all the way to the near Proterozoic glaciations. This is why we call it the boring billing. Don't need to know the details, but basically nothing apparently is really happening. So how is this tied to O2? Well, imagine this. You've got a cyanobacteria that takes CO2, water, and forms organic carbon and O2. If you've got aerobic respiration right beside it, you basically consume that organic carbon and the O2, right? So there's a net gain of zero. Now imagine you've produced organic carbon, but you bury it quickly so that the O2 can't get to it. That means O2 accumulates. So the more organic carbon you bury, the more O2 accumulate in the atmosphere. So that's kind of, I'm going to revisit the vulnerability because this is something else that I work on. But is everyone kind of clear on the general trend of O2? Very low before 2.5, suddenly in the atmosphere, we've got it. We all good with that? So what do I bring to the party? Well, I'm not a microbiologist. I've dabbled, but it doesn't mean much. I work on these rocks called banded iron formations. So this is where all the iron that we use today, we mine from these rocks. So some of you might have seen these stripy colored rocks. These are like the iron minerals, the red and the black. The white is basically quartz. Okay, so they're iron and silica rich. And I want to make a point here. They're chemical sediments. As a non-geologist, what that basically means is that a chemical sediment is something that precipitates out of seawater like a salt versus a detrital thing like a sand, a silt, mud that gets carried by a river. Okay, so band iron formations are largely salts. So if you take a look at the composition of the salt, you can say something about the composition of seawater from which it precipitated. Okay, and the primary minerals are probably things like ferric hydroxide, which you may or may not know. That's basically just iron oxides. It's rust. Okay, so that's the ready colored stuff. The general mechanism for how these things form. So these iron formations form on the continental shelf. Is that a term everyone knows, right? It's when you look at your continent, the water is basically up to about 150 meters deep until you get about 60 kilometers out, and it drops the slope, and then you get the deep sea. Okay, so the shelf is where all the action is. Okay, and it sits on top of actually continental crust. It gets preserved through time. The deep sea stuff never gets preserved because it gets subducted and gets basically destroyed. Okay, so we're forming on the shelf. The theory is we've got hydrothermal activity. The ocean's anoxic. There's no O2. That ferrous iron coming out of the mid-ocean ridges is basically carried by currents up onto the shelf where it gets oxidized to ferrihydrite, forming the red stuff, which is the banded iron formation. And the mechanisms are essentially, I would argue, biological. So back in 2002, when I was first starting out, I was working on iron for well, first time I've worked on iron formation. I was working at hot springs in Iceland and noticed all the iron and silica coming out. And I remember writing this paper saying that basically hot springs are like analogs for iron formations, doing the mistake that most junior people do, like arm waving about things I knew nothing about. So I knew nothing about band iron formations. People point out I knew nothing about band iron formations, so I thought maybe I should learn something about band iron formations. So I did. And then I wrote this paper saying, how many bacteria would it take to form a band iron formation? Because as a biologist, you'll appreciate this, but as a geologist, when I was giving talks in the early 2000s, talking about the role of microbes in geology, geologists just looked at you and thought you were just nuts. I mean, how can something you can't see form these big, massive deposits? I mean, everybody in geology pretty much now accepts the fact that microbes play a role because they precipitate things, are in abundance, right? They metabolize. And if you look at a thick deposit, you have to kind of deconvolute it to an annual deposit. So I can look at something that's hundreds of meters thick, but if something like only a millimeter a year is being deposited over the course of millions of years, obviously you get a thick deposit, right? So I was one of the first to propose that bacteria could do that and kind of taking traction. And the way it does it is one of three ways. So at some point, we'll start with the more recent one, cyanobacteria. Okay, cyanobacteria evolve, they produce oxygen. That ferrous iron, which is being upwelled, reacts with oxygen and forms ferrihydrite. So the cyanobacteria, the only thing they're doing is metabolizing. They're producing that. Now inorganically reacts with the ferrous iron in the ocean and it forms ferrihydrite. At some point in time, you're all familiar with the term cumulative autotroph? Okay. I just assume you do, but when I do that to geologists, they just look at me like, say what? So anyways, the things like gallium-nitride, right? At some point in time, organisms learned to utilize that metabolism because, oxidizing ferric iron with O2, the energetics are pretty bad, right? So they have to work super hard to do this. But at some point in time, they evolve, they can do it. And then there's what we call the photoferrotrophs. So that's basically anoxygenic photosynthesizers. They're not the ones who do anything with O2. They use things like ferrous iron. Okay, so they oxidize ferric iron. They grow below the cyanobacteria because oxygen is toxic to them. Now, in terms of evolution, these guys came first. Most people believe anoxygenic photosynthesis precedes oxygenic photosynthesis. Then comes these, and then comes these. Irrespectively, you've got a biological system oxidizing ferrous iron to ferric iron. The cells in the ferric iron then settle to the sea floor. They get buried, and then they undergo something called diagenesis. That's basically converting your sediment into a sedimentary rock. So basically, the carbon is an electron donor for all sorts of things, right? Your ferric iron is an electron acceptor, so you get the and through that you produce ferrous iron. You can produce any one of these other minerals. These are the main minerals we form in bandite formation. So if I were to give a talk just on iron formation, I would make the argument that it's almost an entirely biological process, from the precipitation of the ferric iron to the re-reduction to form things like ferrous minerals. Okay, so it's all biological. And like I said, 20 years ago, trying to explain this to geologists that this was biological was met with skepticism, but now it's kind of given. We all good with what iron formation is? Okay. Let me explain to you then some of the things that how I can use it. So back in time, there was this paper by a colleague of mine who looked at iron formations through time, and he looked at the chromium isotope fractionation. Now, again, the specifics are not something I'm just gonna explain them to you, but you're not going to be tested on. I'm not going to ask you some question like this. Just so more along, just follow along what I was saying and get the gist, right? So what the argument is this. He looked at chromium isotopes in iron formations through time, and he observed a bunch of positive fractionations. So here's the essential model. In the rock, like let's go back to something like Hawaii, where you look at these dark colored rocks. There's these minerals called chromite, where you've got chromium in it. That chromium is +3. It's insoluble. Okay, so chromium-3 is insoluble, but it can be oxidized to chromate, which is soluble anion, +6 through O2. But O2 doesn't do it directly. It does it through this really convoluted system where O2 oxidizes manganese-2 to manganese oxide oxidizes chromium-3 to chromate. That chromate being soluble dissolves into groundwater, to rivers, and gets carried to the ocean. That oxidation step gives you a positive fractionation. That chromium-6 now hits the ocean, where it instantaneously reacts with ferrous iron, which we know we've got lots of because we have iron formations, re-reducing it to chromium-3, which gets buried into the iron formation. This is quantitative. And what I mean by quantitative, it means even though there's a fractionation associated with it, because all of it gets reduced, there is no net fractionation. So the fractionation that gets carried into the rock comes from this step right up here, which gives you a positive fractionation. All you need to know is this. According to this paper, whenever you see a positive fractionation, that tells you there must have been O2 around. So they've got O2 all the way at 2.8 billion years. Now this is big. So this gets into Nature, which you know is like a really good journal, right? It gets into Nature because they're saying the GOE is at 2.5, but I've got evidence in my rock record that O2 was around at 2.8. So that's kind of why they got into this paper. Now, my group was intrigued by that. We don't work on isotopes, but we work on trace metal concentrations. So what we did is we took a look at iron formations through time. And in terms of the color scheme, it's not really that important, but basically the reds are the band iron formations. The blue are something slightly different. Remember I said I like band iron formations because they're like salt, right? But sometimes these iron formations form really near shore, which means that stuff from the continents is being washed into it. We call those granular iron formations, or what I call the dirty BIF. Okay, I like to look at the composition of the BIF salts because they give me something about bulk seawater. But if I want to know something about continental weathering, I look at the stuff that forms near shore. That's these, Jeff, that's the blue. And essentially what we see is chromium concentrations ramp up at 2.48. They peak at 2.32, and then they drop. Okay? Once again, the numbers are irrelevant here. But here's the thing. Why do we get this peak at 2.32 and then we never see it again if the first model is right that all you need is O2? As soon as you get O2, you get chromium mobilization as chromate. We should see a continuously high amount, but we don't. So we're thinking that something else is at play here that's not just the simple oxidizing chromium-3 to chromium-6. Now, if you take a look at a speciation plot, you could do this for any chemical, but basically, this is showing you the EH-pH conditions. Everything under this line is chromium-3. Everything above this line is chromium-6. So that Fry model was predicated on forming this stuff here, high redox potential. But you can solubilize chromium as well under low pH conditions. You know, great example of low pH conditions if you want to study it, acid mine drainage. And as we all know, in acid mine drainage, pyrite oxidation can happen abiologically, but you wouldn't get acid mine drainage if you didn't have biology, the bacteria, the chemolithic autotrophs, oxidizing the iron and the sulfide. So we write this paper in Nature following on from that saying, well, we don't think it's chromium-3 being oxidized to chromate. We're thinking instead it's chromium-3 being dissolved because of acid being washed into the oceans. It's still an O2 story, but the nuance is very different. So I'll get back to the why it's a one time only. So I'm trying to picture here a landscape of what did Earth look like before there's plants. I'm trying to show it, okay, excluding the plants there. But it looks barren. It's really acidic. Imagine you've all seen it. Have anyone seen an acid mine drain or acid mine before? You know, you just look at the orange, yellow discoloration, stuff like that, right? So imagine a really bleak landscape where everything's been acid weathered. That's kind of the vibe I'm giving. Here's our land. Here's our oxidation on land. So basically, here's my argument. The chromium-3 that we see for sure because we measured it, right? Those peaks at 2.48 to 2.32. I'm saying it comes in as chromium-3, not as chromium-6, and it's coming in with acid. Here's why. The land, up to this point in time, has had no O2. We've got pyrite sitting on land for many hundreds of millions of years, not being oxidized because there's no O2 to oxidize. Suddenly, there's O2. We get the evolution of the chemolithic autotrophs, the aerobic ones. They oxidize to pyrite. Pyrite oxidation gives you acid. That acid today is what you measure, plus it also dissolves sulfur and iron. But that acid would also dissolve any other minerals around, like the chromium, putting chromium-3 into solution, carrying it into the ocean. So my argument is the chromium that we see in the iron formation came in from this big acid pulse. Now, here's the cool story why we got this into nature. Is because we've argued one, requires chemolithic autotrophs, that's 2.48, so we can put some kind of a timeline on their evolution for aerobes on land. But it also explains the one-off. Because prior to the O2, pyrite's stable. Suddenly that pyrite meets oxygen, meets the bacteria, it all oxidizes. As we oxidize all that pyrite, we get this big acid flush into the oceans. But now all the pyrite's been oxidized. So now the atmosphere and the crust are in equilibrium. There is no more pyrite flush. So this was a one-off in Earth's history where we had this big acid drainage, a natural acid drainage, not because of mines, but simply because the pyrite on land has, for the very first time ever, seen O2. So that's why we have that peak then and we never see it again because it was only a one-off. So in our paper, we basically sum up and said, okay, so we have pyrite oxidation. It brings in chromium, but in our final paragraph, now the thing is when you write these type of nature science papers, your final paragraph always has to be the big arm waving, I've discovered God type of stuff, right? So what we basically said is, okay, you've got chromium being dissolved. Either for paper couldn't care less about chromium. But when you start milking it and when you're saying, well, if you're going to dissolve chromium, you can dissolve apatite and phosphorus. If you can dissolve phosphorus and bring it to the oceans, you're going to increase primary productivity. If you increase primary productivity, you bury a lot of organic carbon. You bury a lot of organic carbon, you put O2 into the atmosphere. That's your Lomagundi event. So that's kind of our argument for how this all links together. The thing I do in my personal research, why I call myself a geobiologist or a geomicrobiologist, is because for me, everything about ancient Earth, as today, it's about the link between life and the environment. Right? Geologists just like to study the rock cycle. A lot of microbiologists just like to study the life. It's the interaction of both because everything on the Earth involves both. Okay? The environment changes the microbes change the environment. Cyanobacteria is a perfect example. No cyanobacteria, no O2. No O2, no oxidative weathering. No oxidative weathering, no flush of nutrients to the ocean so that cyanobacteria can be increased. It's kind of like the Gaia hypothesis to some extent here, right? And the cyanobacteria creating their own, promoting their own growth. It's a positive feedback because the O2 they produce creates more weathering on land, puts more stuff into the oceans for them. That'll make kind of sense. So our paper was followed up by another paper that talks about this oxygen overshoot. That's when I said that O2 might have gone up to the modern level. Right? Now, not everybody agrees with this. Some of us do. Now, in the paper, my paper and in this paper, they basically said, look, phosphate is the key nutrient for primary productivity through time. You probably knew that, right? At least in the modern. So the question is how does phosphate from land get to the ocean? All the papers like to assume it's basically dissolved phosphate. But in reality, that's not the case. Today, if you looked at a river and you looked at what phosphate there was, most of it's actually transport and suspension in terms of organic carbon, in terms of iron aluminum oxide, or on clays. So like I said, very little phosphates in solution. Now, in terms of what would have been around at 2 billion years ago, there would have been some organic carbon as microbial mats on land, but there's no plants. We'd have had iron aluminum oxides at some point, but if I've got acid weathering, I can't have ferric iron because that dissolves. Aluminum oxides for sure. If you went to a place like this, just going up on a tangent, if you went today to a place that's really weathered, what you would find in the soil and stuff are things like aluminum. Like, has everyone heard of gibbsite? Basically, like laterites, stuff like that. These are really, if you think in the tropics, the soils, the ones that are really red, yellow colored, that's iron oxide, aluminum oxide, and quartz. Everything else has been leached away. Okay, so you'd have lots of aluminum oxides. They can transport phosphate, and so can clays. So I had a PhD student, Wadu, who came up with this hypothesis for how do we transport the phosphate from land to the oceans, trying to link it to the Loma Gundi cycle. And what we did is we took a bunch of different kinds of clays. So I don't go into details of clays, but kaolinite is, as an example, is one of those clays that's found in really highly weathered things. Kaolinite is what you make porcelain from, stuff like that. So it's the really weathered clay. And what we did is we basically just create these isotherms where we took an amount of clay and added phosphate at different pH conditions. You can see 4, 6, 8 in different ionic strengths, fresh water and marine water or seawater. And what one can see is that in terms of the amount of phosphate that binds, you can bind more phosphates to your clay, low pH conditions, than you can at high pH conditions. Anyone want to hazard a guess why that would be? Anyone know anything about phosphate speciation or any kind of? Right. So has everyone heard of the term deprotonation? Right? Like, so you got H3PO4 that becomes H2PO4 minus, right? You lose a proton, right? Phosphoric acid deprotonates around 2.5 pH, which means now it's a phosphate anion plus a proton. Clays at low pH are protonated. They have a positive charge. So you got a positively charged clay, a negatively charged phosphate. They stick together brilliantly. At higher pH, the clay also becomes negatively charged. So you have negatively charged phosphate and negatively charged clay. They ain't sticking together. So at low pH, this is why you see a preponderance of phosphate binding, which makes total sense. I'm arguing for an acid weathering environment. Tons of acid. It's breaking everything down, including the rock in the tailite, which is positively charged. Apatite's dissolving because of the acid. That phosphate sticks to the clay. That clay transports it to the ocean. Now, that in of itself means nothing because you could argue in terms of what you really want to talk about is the bioavailability of phosphate coming into the ocean, right? That's why everybody likes to talk about it as being dissolved because dissolved is bioavailable. On a mineral phase, it's not really bioavailable, right? If it's on an aluminum oxide, that phosphate hits the ocean, it just sinks. If it's on with an iron oxide, it hits the ocean, it sinks. It's only when the iron gets reduced during diagenesis that the phosphate gets re-released. But that could take millions of years. Okay? What you need is something that drops it quickly so it's bioavailable. So what we did is we tested the bioavailability by something that we call these active experiments. So we had a beaker where we had our clays, our kaolinite loaded up with phosphate. And then what we did is into that beaker, we increased the alkalinity. So we brought up the pH, we increased the salinity as well to make it into basically a marine system. And what you see with time is that the aluminum oxide, the iron oxide really don't release any phosphate as expected, but the kaolinite does. Why? Because again, remember here, kaolinite at low pH binds a lot, at high pH it don't, right? So if you're carrying a particle where the phosphate absorbed on land at pH 3 and it's getting carried to the oceans, suddenly hits pH 8, that phosphate's going to come off. So our proposal in this paper was this is the source of bioavailable phosphates for that primary productivity. And now we're getting into the pure microbiology. So I've got another student who, Yuhao Li, who said, why don't we grow some cyanobacteria and just see if that phosphate's bioavailable? Bring in my lab as well. And what we basically did is we grew these cells called Staphylococcus. These are primary, these are cyanobacteria. We grew them multiple times, so we basically knew when stationary phase was. And what we basically did is following just the gray line here. Our guys are growing along happily stationary phase, and then they go into death phase, right? Like any kind of growth phase, right? They're down here death phase because they've run out of phosphate, right? All their dead buddies have sunk, taken the phosphate out. What we did at stationary phase then is we did two splits where we took some of the cyanobacteria and we added phosphate, but we didn't add phosphate. Instead, we add the phosphate tied to kaolinite. So we added clay with phosphate on it and relied on that phosphate to desorb from those clay minerals. And what you see is the ones with a little bit of clay and with more clay, the populations bounce back. In other words, it's a proof of concept that the phosphate that's being delivered by clay is bioavailable. Because again, the bioavailability is the key here, right? Bringing in phosphate that's not bioavailable does nobody any good. Okay? So, a really simple question. Can a river sediment plume lead to enhanced primary productivity? And I say yes. So you can see how I'm trying to bring this all into big picture, linking the geology and all that. So we're now after Lomaguni event, which ends about 2 billion years. And I'll leave you with this point right here. I'm not leaving you at this point as though the lecture is over. I'm leaving you with this current thought. All right. So if high O2 results from enhanced primary productivity, you can all agree. What does low O2 in a rock record mean? It means, especially if cyanobacteria evolved around 3 billion years, is that one, there are too many things that scrubbed out the O2 that was originally produced, or the cyanobacteria didn't produce a lot of O2. What I'm basically saying is this. Great oxidation events at 2.5 billion years. That's when O2 accumulates in the atmosphere. It doesn't tell me when cyanobacteria evolved. Now, molecular clocks tell us they evolved around 3.5. We've got geochemical evidence saying it's around 3 billion years. The question then is, if cyanobacteria evolved at 3 billion, why does it take 500 million years for O2 to accumulate in the atmosphere? Cyanobacteria produce rapidly, right? They drive primary productivity, right? Our O2 cycle, like half of the O2 that we breathe comes through cyanobacteria. The other half is terrestrial plants, right? So it's a big time producer. So if they evolved early, why did O2 in the atmosphere not accumulate early? So that's kind of the question I'm going to basically try and resolve. Let that to 150. We're going to get that way before that. Am I speaking too fast? No, I'm almost feeling winded here. I'm tiring myself out. And I've only gone halfway. All right. Take a breather. What is the evidence for O2? Physically, the evidence is lousy. It seriously is. People have argued we've got these microfossils because they look like cyanobacteria. We've got stromatolites. These are microbialites that are fossilized. They could be cyanobacteria, but there's no smoking gun really for cyanobacteria. These things are some of the closest things, at least in my mind. These are these things called microbially induced sedimentary structures. This is a modern microbial mat, I think from Tunisia. Yeah. You know, you've seen this before. You walk out into an estuary, you know, the gloopy, not only just gloopy mud, but it's all the cyanobacteria and the algae growing on top, right? So you get this texture. This is what the authors of this paper think this texture comes from. It comes from something growing as a microbial mat. Here's the thing. Most microbial mats today are cyanobacterial. So the argument's always been if you see a microbial mat in a rock record, it's probably cyanobacterial. But we don't know that's true because there's anoptogenic photosynthesis. We certainly know when you look at, I don't have this slide here. Has any of you ever seen like a picture like of a microbial mat when you cut it perpendicular, you see the different colors, like you see the green, you see the purple, right? The green's the cyanobacteria, the algae, the purple's the purple bacteria, then you've got sulfur bacteria under and sulfate reducer at the bottom, right? It's laminate. So we do know that anoxygenic bacteria can form laminations. Can they form a mat all on their own? Well, they don't do it very often today, but microbial mats are pretty rare today. And just out of curiosity, why don't I walk everywhere and find a microbial mat or a stromat, something like this? Why is that more rare than because most of the time, I mean, Tunisia here, most of the time they're formed in evaporative environments, like you think of Persian Gulf. They're formed at hot springs. Am I, are we getting closer to why we don't get them everywhere? No. If you're an animal, you love plus 70 degrees. You love hypersaline conditions? Probably not, right? So animals are limited. You limit animals, they grow. In the past, before animals, the entire Earth would have looked like this. Now with animals, basically these things only exist where basically animals are excluded. Okay. Now, some geochemical evidence, because a lot of the stuff, evidence for early life comes from geochemists. And I always laugh about it that the microbiologists have fallen behind here because the geochemists are driving the narrative of evolution more so than the microbiologists are, because we're the ones with the rocks, and you're not. You're the ones with a culture and a test tube. So there you go. Anyways, so what is was that? So you're gonna have to believe us, even though we're just making this stuff up. Yeah. But this is our information from 3 billion years ago, and this is a study was on where we're looking at molybdenum isotopes. So this is just one of many different stable isotopes you can do. And when you take a look at when you compare the molybdenum isotopes, which none of this will mean anything to you now, but I'll just tell you what the main point is, versus iron manganese ratios. What you find is a lot of these things that are high manganese down here have very low fractionations. Now, the only way you can get a fractionation that low is if you have something like a manganese oxide absorbing molybdenum from solution. That gives you a really low light fractionation. In the absence of the manganese, you get fractionations that are up here. What I'm basically getting at is if you look at the rock record and you find, A, you've got a manganese rich rock, and you've got this high fractionation, this low fractionation, this tells you you must have had manganese oxides. Why is that important? Because to get a manganese oxide, you need abiologically, this reaction is super slow. At anything without O2, it's even slower. Only hydrogen peroxide really works. Okay. Find a manganese oxide tells you you must have had O2 and probably had life, but it can't happen just abiotically. So the argument we made is if you go to these rocks all the way 3 billion years ago, we must have had O2 around to give us the manganese oxide that could then give us this molybdenum isotope fractionation in these rocks. Okay? You could be rightly looking at that and saying that seems like a bit of a tenuous connection, and it is. It's the best we can do, right? All we can do is we can say we've got this fractionation. What are the mechanisms of getting it? And the only one we know of really is through manganese oxides. So the question then is, how do I get a manganese oxide? Need O2. Okay? So let's accept that at 3 billion years ago, we've got O2. Let's accept that great oxidation event's at 2.5. We've got a 500 million year gap. And the question is, why? So one of it is there's just too many things that we're reducing at the time. So we have an atmosphere that's dominated by things like CO2 and methane. Now, how do we know that we had a lot of CO2 and methane? If you look at the trajectory of Earth's cooling, for example, at about 4 billion years, whatever like that, the temperature is about 1,500 degrees on the surface because everything's molten. By the time you get to 3.8, you've already got things like rocks, like continents, which tells the temperature is at least 1,200 degrees. This is based on what we call the Bowen's reaction series in terms of how you basically crystallize minerals from a magma. Okay, then you've got water at 3.8, which it tells you that so at 4, we're at like 1,500. By the time we get to 3.8, we're at less than 100. Based on that trajectory, we should have been in a deep ice age for a long period of time, but we weren't. First glaciation is at 2.4. So something kept the planet warm. The geophysicists who do the modeling on this say we must have had a lot of greenhouse gas, like 100 atmospheres of CO2 and a lot of methane. So if we have a lot of methane, we can't have a lot of O2, right? Those things are incompatible. So you can argue there's a lot of methane. All the crust, all the volcanic rocks are ferrous iron containing, not ferric iron containing, like the basalts. So they're a source. The oceans have a lot of iron. There's a source. Volcanoes are giving off H2, H2S. So the question is there so much of this reduced stuff around that it takes 500 million years to scrub all the O2 out? That's a possibility. So I wrote a paper way back when looking at in terms of the methane story. So this didn't start off as so many research projects are. It starts off as one thing and it kind of morphs itself into something completely different. This project started off with me having a PhD student who analyzed these rocks. So I don't know why all the time it converts it, I always use the Nazis. This is basically 2.5, that's 4, that's moderate. Okay, so that's 2.5 the GOE. That's concentration of nickel. He basically did his paper, he said 2.5, we find all this nickel and iron formation. I thought, okay, that makes sense. We got oxygen on land, weathers oxidizes stuff, brings the nickel in. So the thought was, if I go back into the rock record, I should see low concentrations versus this. We see absolutely the opposite. We see instead super high concentrations and then it drops. So why is that the case? Well, what we argued is that this reflects basically changing temperature on Earth. And here's why. The early Earth is a lot hotter than today, right? Because right after the collision, you've got all that heat, right? You've got radioactive decay, so the planet's super hot. That means we got more partial melting of the mantle. The mantle is made up of rocks that are rich in magnesium, iron, and nickel. So as long as there's lots of partial melting, the planet's hotter, the volcanoes that erupt have more nickel in them. As soon as the planet cools by about 2.5, they don't erupt as much of the mafic material anymore. They become things like basalt instead like Hawaii. There's less nickel. So what we argue is that this trend directly mimics the temperature change of the planet. In fact, these are just the predicted temperatures of lavas coming out at that time. This is the modern down here. You can see like 1200 degrees versus 1600 degrees. In other words, we had a super hot planet. That super hot planet mantle was melting. The volcanoes which erupted, lavas, had a lot more nickel. That nickel erupts into the oceans, it dissolves, it gets captured in banded iron formations. So we're following this trend here. But again, as far as a top tier journalist, it's like, whatever, that sounds like a very geology story, couldn't care less. So we started thinking, okay, what is the link to biology? Now, if I talk about nickel, what enzymes do you think are important? Hydrogenase and? You're absolutely right. There's a certain group of bacteria that need nickel more than anybody else. You begin with an M, followed by E, methanogens. With that, they're archaea. The genus is methano something. Yeah. So, anyways, methanogens. Okay. They need nickel more than anyone else. So the thought was, okay, if nickel's high and then nickel's low, does this mean anything for the biosphere? And it kind of makes sense because if methanogens, oops, sorry, I got rid of that slide. Never mind. If methanogens need nickel, then they're super happy early on, right? Now we know they're super happy early on because the planet is warm and it's high in methane. But we know then that the great oxidation event represents a tipping point where O2 exceeds methane in the atmosphere. So what causes that change in the tipping point? Because you can't have high both, right? Early on, high methane, low O2. At 2.5, it's like this. Okay, so something's happened. But people have made the argument before that cyanobacteria evolve, they produce O2, it strips the methane out. Our take on the story is it's got nothing to do with the cyanobacteria, they're incidental. It's instead the methanogens are super happy early on, as long as there's lots of nickel. The nickel drops, they starve, they produce lots of methane. As they produce lots of methane, the cyanobacteria that are in existence at that time simply outcompete them. So that's our tipping point. So that's how we got this paper in, basically saying intellectual thing. Why I love this story, and I particularly love this story telling it to geologists, because to me, it represents the whole Earth as an integrated system. We start off with a hot planet, purely geological. It changes to volcanism, purely geological. Those rocks now dissolve in the ocean, right? So there's high nickel in the ocean versus not, abiological. But that now affects the microbiology, which affects the methanogens, which affects the gases, which get emitted, which then affects the redox state of the planet. See how we've gone full circle here. The microbes are a part, the geology is a part. It's the way everything interacts together, okay? So the question then becomes, is there any proof of any of this? And I think there actually is. So this came from a paper a few years back looking at organic geochemistry. So they're looking at rocks over all this period of time from 2.5 to 2.7. So it's a continuous sequence of rocks from Australia. And what these symbols basically represent, the circles everywhere represent shallow rocks, the triangles represent, and the squares represent deep rocks, deep water rocks. So shallow being near shore, further away from shore, okay? And they're looking at the organic carbon, the kerogen that's preserved in these rocks. So they're looking at things like shales, carbon. So they extract the organic carbon. They do basically the carbon isotopes on that organic carbon. What you see when you look at 2.7 is it doesn't matter if you're looking at shallow water or deep water, the isotopic fractionation is pretty high. Now that isotopic fractionate, cyanobacteria fractionate up to around here. The purples, the greens, they fractionate over to here. What fractionates that high? What group of species? Begins with them, all by me. There we go. Yeah. So methanogens, so it's methane cycling. Methanogens produce isotopically like methane. Methanogens, the ones that can oxidize them, right? Methane, basically become enriched then in very light organic carbon. So basically, methane cycling is taking place at this point in time in deep water and shallow water. Now, what this study got into PNAS, what their argument was, is that methanotrophs today, if you think about what oxidizes methane, it's O2, aerobic, right? Sulfate, manganese, nitrate, and iron. Aside from iron, which can be or can be done without O2, the rest of them are O2 storing because you don't get sulfate unless you oxidize pyrite. You don't get nitrate unless you oxidize ammonia. You don't get manganese 4 unless you oxidize manganese 2 with O2. No matter how you cut it, there must have been O2 around to give you the signature for methanotrophs. But here's what I think relates to my story. Everything deep, so methanogens are living in shallow water, they're living in deep water. They're basically ruling the roost in the oceans, right? By the time we get to up here 2.6, the methanogens are still happening in deep water, but the shallow water is now dominated by cyanobacteria. So what I'm arguing is that after 2.7, when methanogens begin to starve, as they starve, they contract. Cyanobacteria that are already living in the shallow water are now basically out competing and they're taking over. Now, another thing that happens, which I haven't talked about at 2.7, is the continents get bigger. Early Earth was dominated by just volcanic islands. Think a bunch of Hawaiis, that's all you had. And through time, these islands get bigger, 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 you get more bigger continents, okay? At 2.7 to 2.5, the continents are getting bigger. The implication of continents getting bigger, suddenly we've got more continental shelf. Continental shelf is that shallow water that's basically colonized by benthic mats and by plankton. So imagine if you just had a Hawaii and you were colonized by benthic mats. You'd have a very thin fringe of cyanobacterial mats, right? Because you got a steep volcano going into water very quickly, right? It means slope like that. If you have a shallow shelf extending out, let's say 20, 30 kilometers, that's a lot of habitable space for cyanobacteria. So the cyanobacteria are taking over there, methanogens are marginalized. Now, what's really cool is, where do I find methanogens today? And I'm not talking about in your gut, in terms of the environment. I don't know if someone said something. Okay, let's get rid of animal life. Let's just think in terms of environment. Where would I go today and say, find me an environment? It could be high temperature environments. If I went to the beach, right, and I were to dig the sediment away, would I find methanogens at the surface? No, I'd find aerobic respired, right? Has everyone done like sediment biogeochemical zones, like oxic, suboxic, anoxic? Oxic is basically O2 at the top, and you've got things like nitrate, and you got manganese reduction, iron reduction, sulfate reduction, methanogenesis. Basically because O2 is a better electron acceptor, more energetic, right? Followed by nitrate, manganese, iron, sulfate, CO2. Methanogens are buried in deeper sediment. So what I'm saying is once upon a time, they were everywhere. Now cyanobacteria marginalized them from shallow water, so they're just deep water. After the GOE, though, suddenly we have pyrite oxidation, because I talked about that. Higher oxidation gives you acid, but it also gives you lots of sulfate. Sulfate in the oceans gives you sulfate reduction. The sulfate reducers basically push the methanogens out of the water column altogether and marginalize them. So once again, everything's linked together. And the second piece of evidence are these things right here. These are our stromatolites. In terms of abundance, they really pick up after 2.7. Expect there's more continental shelf, there's more habitable space. These microbial mats are getting bigger. So it's a combination of one starving, the other one having more land to expand. So as one does better, one this is like every day, right? I mean, something suffers while something else does better, right? So that's I think the story what's happening. Now I've got a slight problem. Checking my time. I've got one slight problem. People have done these calculations and said, look, if you take modern O2 production of microbial matter and you were to integrate, let's say if you were to take, so this is like just a study we're doing right now versus someone else's. Imagine I've got one singular continent, let's say a tenth of the land mass exposed to today. Okay. And just to put numbers on this, before 2.7 billion years, that's kind of what we're looking at. It's 10% of the continental land mass. After that, we get bigger. And if you were to say microbial mats were growing all the way around this continent, let's say up to 10 meters deep, producing O2 at modern O2 rates, how long would it take to burn through all those reductants? And the answer is thousands of years. Here's a problem, right? We're arguing that 500 million years. The calculations show that it should be super fast. And indeed, I've really created a problem for myself because I thought I was being dead clever years ago when I did a study with Eastern of mine looking at microbial mats on land. And in fact, that's what we did. We measured O2 production rates. And never mind, I'm too much detail. What I basically said is that if the land mass, if modern continental land mass had only 1% of it covered by microbial mats, it would be all the O2 we need in our atmosphere today. Okay? Because microbial mats are super productive at producing O2, right? They're tightly packed in there together, right? What was the equivalent? Something like 100 meters of water column, the same as a centimeter of a microbial mat from O2 production. Okay? So we quickly kind of saying, well, look, if cyanobacteria were super abundant and happy, they should have oxidized everything. That 500 million year gap can't be due to that. But here's the problem. If you go out into the modern world, we don't have this nice green lawn of microbial mats going everywhere. And my final slide is, to me, what I think is the key to all this is actually the evolution of what cyanobacteria. The cyanobacteria we look at today, like the Anabina, the Calophyrex, and all these things, weren't around at that point in time. So the argument I'm going to end with is that we didn't have the right cyanobacteria. Even just from a geospatial distribution, cyanobacterial mats aren't everywhere. So perhaps they simply were producing O2, but there wasn't enough of them. And then to me, what I'd like to describe is the elephant in the room for everybody in my community, because nobody wants to talk about aerobic respiration because the general premise is aerobic respiration occurs after the great oxidation event when O2 is abundant. Again, this is driven by geologists, not by microbiologists, because we see everything as macroscopic. And as I try to explain to my biology colleagues, when a microbe's only 1 micron, you got 1,000 lined up to be 1 millimeter. Imagine 1,000 of us lined up in a row. I'm on this end. I don't even know who's over there, right? Geologists, we don't tend to see the world like that. We see mountains, right? You see microscopic. So for me, it's kind of like, at what point in time do we start getting things like aerobic respiration, even though, we tend to think of in broad terms, like what would you physically see? Now, this paper right here came out actually arguing based on molecular clocks that oxidases already existed by 3 billion years. So you have enzymes that today serve as terminal oxidases. Whether they served as terminal oxidases back in the past, who knows? But the real question is, if aerobic respiration comes soon after cyanobacteria, they're going to hold each other in check. If there's a big gap, then we got a problem. So this to me is one of the big unknowns. We just don't know when aerobic respiration really kicked in, because what would we actually look for as a fossil in the rock record to say these guys exist? We can't. So the second thing then is things like growth limitations. Now, everybody in the geochemistry community likes to talk about phosphate being the key limiting nutrient for everything. Okay? And a lot of this work comes from this classic paper right here in 2002, where Bergstrom and Canfield, so Down Canfield is like a super big name in the geochemistry world. And what they did is they looked at these black smokers and they looked at the composition of iron and phosphate. So the phosphate iron ratios in the solids related to dissolved phosphate in the ocean. And when you plot that, you get basically a partitioning coefficient. In other words, the amount of phosphate tied up in the particles is directly related to how much phosphate's in solution. Kind of makes sense, right? The more phosphate you have in solution, the more you potentially bind. Now, if you then plot this is iron formations. This is the phosphorus iron ratio and iron formations through time. There's obviously a variation because there's a variation in the rocks through time. Where this bar, orange bar, intersects the red line at the top and at the bottom, that is the equilibrium concentration of phosphate that must have been in seawater at the time that these things were forming. And here's the point. They argued that you had only about 0.15 micromolar compared to today, which is about 2.3. Again, don't worry about the numbers. Formations were forming, and all this iron sinking out of the water column. Iron is a great, basically, it's a sponge for things like phosphate. It's sucking up all the phosphate. As it sucks up the phosphate and the iron settles, it's taking phosphate from the water column, particularly the photic zone. If you're in the photic zone like a cyanobacteria, you need phosphate, you've just said goodbye to your phosphate because of the iron. So as long as iron formations were forming, the oceans were limited in phosphate. So, fantastic story. I kind of totally agreed with it. But when you look at iron formation, iron is not the dominant component. Silica is the dominant component in iron formation. Today's oceans have hardly any silica, but in the past we had a ton of silica in the oceans. Any idea why in the past there was a lot of silica, but today there's not? Yeah, precipitation by what? This is in your biology realm. Who takes out silica today? Begins with a D. I. Diatoms. Diatoms, sponges, radiolarians. These things don't evolve until 600, 500, 200 million years. Okay? In other words, early on Earth, there was a lot of silica, but there were no organisms that actually made silica skeletons or shells, right? They evolved much later on. So the oceans had a ton of silica. So the point we made, not going into too much detail, we said, hey, if iron particles are settling out of a water column, yes, they take out a ton of phosphate. So when we do it just phosphate and iron only, we get the exact same thing as Bergen cancel. But when we do high silica, we size over phosphate. In other words, the more silica you have, the less phosphate they have. In other words, we made the argument that there was no phosphate limitation because there was so much silica in the ocean. That's what was getting scrubbed out. That's why you get the iron silicates and iron formations. Then we did a subsequent paper where we looked at iron formations through time. Now, here's why you've got to do the geochemistry, get the KD partitioning thing. If you look at a rock, these are iron formations and these are other iron-rich rocks. And you look at it, you would say the modern stuff has way more phosphate in it than the past. So there was phosphate limitation. In of itself, that's absolutely true. But you have to take into account the chemistry. You can't just look at a rock and its composition and say what was dissolved. You have to do the experiment that says how much stuff dissolved can partition to the primary precipitate to then look at the rock record. So you need that intermediate chemical step. If you do the KD experiments, the stuff that we did, the nitty-gritty stuff, what you find is that the red is basically, what we're finding basically, these numbers, in fact, we do have higher phosphate early on than today. In other words, there was no phosphate limitation. So what happens, just one of these off-tangent things, what happens is when you've written enough papers, you start actually sometimes contradicting yourself without even thinking about it. So we got this other paper we get into Nature, where I'm the lead senior author on this one, saying phosphate through time in shales. So these are basically muds that turn into rock. And what we see is after 700 million years, you get a ton of phosphate, before that's not. So the general premise is that the shale record is showing us a big pulse of phosphate in the modern, not in the past. So I look like I'm contradicting myself here by saying, wait a second, there was a lot of phosphate in the past in the iron formations, but the shales are showing me something completely different. So how do I get the best of both worlds and maintain some consistency? So I'm struggling here. And I think, okay, let's take a look at where these things form. So just basic geology. Here's our land. Here's a river bringing all our sediment. So we get sand, silt, and muds, right? and they get basically ejected by waves. The information is affording itself. The argument that I've been making is that this stuff here tracks what's in the deep ocean coming that's being upwelled. This stuff here is tracking what's coming by rivers. So these things can be fundamentally different, and they should be because you're representing two different environments. So with the informations, I am tracking deep ocean, which has no problem with phosphate, versus this over here that does. And the reason this is low is because there's less continental weather, less phosphate that gets dissolved. Okay, so it's just making a point of sometimes you have to think through what the data set's actually telling you and putting it into perspective. Yeah. And then really, this is just kind of one of these interesting tangents about how science goes. I always tell this to my students. If I write a nature paper saying white, you better write a nature paper saying black, because if you write one saying I was right with white, you're not getting into any good journal, right? By default, if we want to publish good, we have to say that you're all stupid and I'm smarter than you, right? And sometimes we have to say I was stupid and I'm not smarter. So if you be self-consistent. So back in the day, when birds mechanics were talking about it was 10% of the phosphate, and I said, well, no, it's like 100%. So for almost 20 years, we're arguing about 10% difference, 10% versus 100%. Who cares? Now we've got people arguing, talking about things like hydrothermal plumes. Like even today, you can have a deep sea emission of material, and iron can be transported thousands of kilometers because it's held in solution by DOC. But people have made arguments that you have actually things like 100 to 4,000 micromolar versus 2, because we're going backwards in terms of what we know. We all are kind of agreeing with 10%. Now we're within 5 orders of magnitude. It's like science in reverse. It's kind of the more we go on, the less we actually seem to know. So, anyways. So here's one way though I think phosphate could be a problem. And this is something that once again, as you start working more and you start your evolution of your ideas. There's a really good paper by Kip and Stoic in 2017 where he said, look, early in life, before there's O2, and you form organic carbon takes out phosphate, right? And phospholipids, ATP, DNA, stuff like that. If there's no O2, you've lost your main terminal electron acceptor. There's no nitrate, there's no sulfate, there's no manganese. That means a lot of the biomass is going to die, settle to the bottom, and it's not going to be oxidized because you've got no terminal electron acceptors. I just wrote this paper with a colleague of mine at McGill, where we wrote something similar, and we talk about exoenzymes. Everyone knows what exoenzymes are? These are the enzymes. So most heterotrophs can't break down complex material, right? They excrete enzymes that break things down, like peptidases and stuff like that. They break them down into smaller molecules that they can take in, right? You can't take in, something that's a micron in size. There's no pore that's big enough. So you have to have everything small. So our argument was that the early Earth, exoenzymes, because they're pretty complicated things, the ones that we know of, it's very likely that the earliest cells didn't have exoenzymes. So another case could be that you have primary productivity, but the organic carbon basically settles out because you can't break it down and re-release the phosphate. So maybe this is just a purely biological story that the evolution hasn't yet kicked in. But that's our thinking today. Someone's going to write a paper at some point that says exoenzymes are 3.5 or whatever. That's fine. Whatever it is, right? But that's what we're thinking today. Right. I am really close to being done. Okay. So last few slides. One of them is to the landmass. I haven't really talked about this much, but remember I said, think about the world being nothing but a bunch of volcanoes and eventually to get bigger. If you're looking at life colonizing on land, in the shallow water, or even on land, the less land you have, the less colonization you have, right? The less O2 is going to be produced. I'm just going to skip this slide because it's too complicated. There's also physiological problems. We wrote this paper talking about ferrous iron being toxic to cyanobacteria. So we did these experiments with Synechococcus, and we showed that at high, like over 100 micromolar iron, they basically produce too much ROS and they do the Fenton reaction, so not super happy. So what we said is basically if the oceans have a lot of iron, the cyanobacteria held in check. So at periods of time where you've got shallow waters, you can have these lagoons with reef systems and cyanobacteria certainly happy as long as the iron's down here. Then you get a transgression, which means sea level rises, brings iron up. What used to be a happy population is now a very unhappy population. So maybe iron was toxic. My colleague, Michelle Geringer, disagrees with us. She says it's not, that's a problem, but paradoxically, Cyanobacteria simply lack the ability to actually extract iron. So the importers that are used today for bringing iron into the cell hadn't evolved yet. So maybe it's a physiological thing. And then we did a thing about UV protection. You imagine your plankton growing at the surface of the ocean. There's no ozone, which means UVC is penetrating. You're just getting fried. So maybe it's something like that. It'd be something simple as this. You've got small volcanic islands, and volcanic is the key word here. You've got lots of pyroclastic material. You're growing super happy on the coast, but you're constantly getting covered in pyroclastic material. Not good. You got steep volcanoes and little continental shelf, which means that the weathering rates are going to be super high. Imagine being in, I don't know, in Hinton, the speed of the river versus being here. It's going to be a lot faster because of the slope. So you're getting covered. So maybe it's, once again, limiting habitable space. Last two slides, competition. I've talked about the competition between the pathogens and cyanobacteria, but here's another idea that came up. Imagine you've got your purple bacteria, your photoferrotrophs, oxidizing iron, deep in the water here. They can go up to 100 meters deep, which is your photic zone. And then you've got cyanobacteria up here. If the iron is coming up through upwelling, the first species that sees them are the photoferrotrophs. The cyanobacteria don't get the nutrients, the phosphate. They're starving simply because they're growing up near shore in top water. Purples, the photoferrotrophs are growing underneath them. So they get first access. And then why we got this paper in here was another cool little twist on this is at some point when O2 does expand, so the cyanobacteria begin to deepen, and as they deepen, they push the photoferrotrophs back, right, because they can't tolerate O2. But you also oxidize ammonia to nitrate. As you get nitrate reduction, you produce things like nitric oxide. Nitric oxide is toxic to things like the photoferrotrophs. Once again, bringing in the competition. So you can see what I'm trying to do here. It's always not just a geological, but there's also the biological component. Last slide. This is the one I'd really like. So I'm working with this colleague who's a molecular microbiologist in Bristol. And so this is Patricia here. She's worked a lot on the origin of cyanobacteria. And although cyanobacteria, we have a record for them going all the way back to at least 3. You see these things right here, the macrocyanobacteria and the microcyanobacteria. The macrocyanobacteria are the big, thick microbial mats that we find today. The bacteria that formed them, like Nothcalis and things like that, did not exist before 2.5. So the theory is this. Cyanobacteria may have existed, but she terms them the losers because they basically get beaten out eventually by microcyanobacteria. But basically, guys who existed weren't big mat forming. They were just simple filaments like pseudoanabina, stuff like that. They didn't produce much O2. It's only some point in time when this macrocyanobacteria evolved that you get these modern rates of cyanobacterial production. And they come around, once again, it's hard to pinpoint when exactly this is because all molecular clockwork is predicated on something geological. So this can vary, but it's right now around 2.6, 2.5. That would mean that our arguments for how fast O2 can burn through all the reductants could be right. It's simply we didn't have the right cyanobacteria to do it. Okay, I'm going to leave it at that. I know that was a ton of information. But what I really wanted to get out of all this, and I hope you get out of this, is that to understand the way the Earth works, you have to consider the biology and the geology together. They're always linked. One affects the other. And what the kind of the cool science, at least for me, is working at the interface between both. So I dabble a lot in microbiology. Now I'm not a microbiologist, so I've got microbiology colleagues to help me with all this stuff. But it's kind of like the fun thing, working at the interface. But anyways, to at least convey the point that the evolution of O2, you got a broad idea for when it happened, how it happened, and that it's all intimately tied together with geology. We good?
No, I have no knowledge in this area, but I'm loving to hear. I completely agree with you in different perspective. Last time I didn't hear Dr. Konhauser, but thank you so much. I kind of wanted to hear you a lot.
Okay, well.
And that's really thank you. And we recorded Dr. Konhauser, so thank you for agreeing. That was great.